Hello everyone, my name is Tanishka Konda. I'm a high school student from Dallas, Texas, and the main focus of my research paper that I'll be presenting today is, like she said, epigenetic synthetic lethality, and I'll be talking about some of the mechanisms, the dependencies that we can discover in it, and emerging applications and some of the newer technologies that we're using to study this topic. So to begin with, I just want to share a little bit of information about this paper. This is a literature review, which means I studied, analyzed, and compiled anywhere from clinical trials to other literature reviews to really understand and study this topic. And I synthesized these findings to create the paper which I have, and from the clinical trials and reviews, I was able to gather my examples for it. And my goal with this overall paper was to understand how epigenetic changes in cancer cells can be used to expose something called synthetic lethality, which we can then exploit and target for a possible precise way of cancer therapy. And this paper overall just explores this concept of how we can find these examples that we could use to treat cancer. And this overall method would be treating cancer in a way that's selective, and rather than harming all cells in general, and including the healthy ones, maybe this way can offer an approach that we are just able to target the cancerous cells and leave non-cancerous cells alone. So again, I highlight some key examples and technologies, and also some of the growing areas in this field of research. So my paper begins with introducing the concept of epigenetics and answering the question, what is epigenetics? Well, epigenetics refers to basically changes in how genes are either turned on or off. So they'll be expressing different things without changing the DNA sequence itself. So we're not talking about any mutations to the underlying sequence, just overall changes in gene expression. So the two main ways that this can happen is either through DNA methylation and histone modifications. So DNA methylation is where chemical tags are added to DNA to silence certain genes, which will then change expression, as well as histone modifications, which will change how tightly DNA is wrapped around proteins called histones, and this also affects gene accessibility. So epigenetic regulation is essential during a cell's development, and it's going to determine which genes are active in a cell. So this is important because some cancer cells will turn off the genes that are going to prevent this uncontrollable growth, which is bad, and we want to try to control and target this. And abnormal patterns of methylation or histone modification can, again, lead to these incorrect genes being turned on or off, which is something we don't want to happen, which would then help the tumors grow and spread. So the next topic that my paper discusses is why the epigenetics matters in the context of cancer. So cancer, it not only involves mutations in DNA, but will also change how cells are regulated. For example, a tumor cell might silence a gene, again, the example I gave earlier, which normally stops cell division, which is usually called a tumor suppressor gene, or it might activate a gene that makes a cell grow uncontrollably, which would be an oncogene. And what makes epigenetics very interesting is that unlike actual DNA mutations, epigenetic modifications and alterations are reversible, which means that we can target them. So that means if we can find drugs that undo harmful modifications that would help cancer cells grow, we could potentially restore normal gene function and create a healthy cell. And again, this opens up new avenues in cancer treatment, and treatments that often are more specific than normal ones. And then I discuss what is synthetic lethality exactly. So this concept is from genetics, and it means that two genes are only lethal to a cell when they are lost together. And so if either one is working, the cell will survive, but if both are gone from the cell will die. And in cancer therapy, the way that we're going to utilize this is that tumor cells have mutations that will disable certain genes. So if we target their synthetic lethal partner, which they need in order to function, which needs which is what the cell needs to overall function, we can kill only the cancer cells and leave the healthy cells alone. So a famous example of this that has been studied quite often in this field of epigenetics is BRCA1 and BRCA2, which are involved in DNA repair. And in some cancers, such as breast or ovarian cancer, these genes will be mutated, and cells with BRCA mutations will become reliant on a backup pathway or a backup repair enzyme called PARP. And so scientists have developed something called PARP inhibitors, which are drugs that will that will block this backup pathway and will kill only BRCA deficient tumor cells. So this is a famous example of synthetic lethality, and it's already been used in some clinics, but still people are trying to research it more in a clinical setting. And then my paper moves on to talk about three main types of synthetic lethality. We have paralog-based, crosstalk-based, and collateral synthetic lethality. And all of these three types of synthetic lethality will involve epigenetic regulators. So the first one that it discusses is the paralog-based synthetic lethality, where two genes with similar functions, which are referred to as paralog genes, can usually substitute for each other in function. And the second one is crosstalk-based synthetic lethality, which occurs when cancer cells lose one pathway and start relying on the backup pathway, creating a vulnerability that we can target. And the third one is collateral-based, which occurs when a tumor not only loses the key gene that it needs to survive, but also the neighboring passenger gene, which is what it's referred to as, which can also lead to a targetable weakness. And each of these types of synthetic lethality will give us a different way to expose cancer-specific dependencies in a cell that we could possibly target. So a little more about paralog-based synthetic lethality. Paralogs are genes that evolve from the same ancestor and have similar functions. A pretty well-known example is a RID1A and a RID1B, which are part of the chromatin remodeling complex, the SWI/SNF complex. And when a RID1A is mutated in a cell, which is common in ovarian cancer, the tumor becomes dependent on a RID1B. So blocking a RID1B in these cells will kill, in cancer cells will kill them, whereas in normal cells, the cell will remain fine. And another example is the histone acetyltransferases P300 and CBP, and these enzymes help open up chromatin, which is able to turn on or off certain gene expression. And if one of these is lost, the other will become essential, giving us another targetable therapeutic window. And this strategy is important because, again, we want to target cancer-specific weaknesses in cancer cells to try to only target cancer cells while avoiding harm to healthy cells and healthy tissues overall. So the next one is the crosstalk-based synthetic lethality. And the crosstalk-based lethality is about how cancer cells will rewire their epigenetic programs. And so when one chromatin regulator is lost, the cell will try to compensate for this by activating a different yet related pathway. And this compensation can create a new point of failure in the cell if targeted. And an example of this is the enzyme EZH2, which normally represses genes through H3K27 methylation. And if EZH2 is missing, the cancer cell can compensate by relying more heavily on NSD1, a different enzyme. So if you block NSD1 in these cells, specifically in the EZH2 deficient cells, they will not be able to survive. So this interaction is demonstrated in a study named Drozos et al., which shows how tumor cells can become addicted and reliant on this backup pathway. So possibly exploiting the backup pathway will create a therapeutic window again. Moving on to the next form of synthetic lethality, collateral lethality. This will happen during large chromosomal deletions. So sometimes when a tumor deletes a key gene, such as a tumor suppressor, it can also accidentally delete a nearby passenger genes. So these passengers aren't harmful to lose on their own, but their absence may create a vulnerability in some cases. So an example of this is the deletion of MTAP, which is located next to the CDKN2A gene, which is a major tumor suppressor in cells. And in many cancers, both of these are lost together. So MTAP normally helps recycle a molecule called MTA. Without MTAP, the cell can accumulate MTA, which inhibits an enzyme called PRMT5. So these cells then become dependent on PRMT5 to survive. And so blocking PRMT5 can selectively kill cells with MTAP loss. In more recent studies, such as Joe et al., they suggest that similar dependencies might exist at the HDAC1/2 loci, which gives us a new opportunity for, again, these selective therapies. So moving on to another side of this topic, the more technological side, which is what's being used to study this topic. And something that we have to understand is a little bit of background information about how CRISPR works. So to discover synthetic lethal relationships in genes, scientists can use this tool called CRISPR. And CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. And it's a gene editing system, essentially, adapted from bacteria. And it'll let us cut genes and disable specific genes in living cells. And it specifically does that by using a guide RNA to direct the Cas9 enzyme to a specific DNA sequence, and the Cas9 enzyme will then make this cut to that sequence. And this either destroys the gene or causes it to malfunction, which is essentially a genetic knockout. And this technology, CRISPR, allows researchers to test gene by gene on an individual basis which genes are essential in a cancer cell with specific mutations. So there are three types of technologies that are being utilized to identify synthetic lethal interactions. The first one is the CRISPR knockout screen. So the CRISPR knockout screen allows researchers to systematically disrupt individual genes one at a time in thousands of different cancer cells. And again, researchers are looking for which knockouts are only lethal in certain cancer types. And the second one is the codependency screen. And these use large genetic data sets to find, again, gene essentiality. So genes that function together, that's what we want to target. And moving on to the dual CRISPR screens, these knock out two genes at the same time to look for combinations that are lethal. And specifically, dual CRISPR screens are helpful in identifying paralog relationships or backup pathways that aren't obvious from single-cell gene studies. So how can we apply this concept and these technologies to epigenetic targets? When we apply these technologies, we can then uncover cancer-specific vulnerabilities, which is the overall goal. And a prominent example of this is ASH2L, which is a chromatin factor that was found to be essential in glioblastoma cells through CRISPR knockout screens. And on the other hand, a codependency screen allowed us to figure out that there was a strong interaction between EZH2 and MLL-M1, which are two epigenetic complexes. And then the dual CRISPR screens allowed us to confirm that deleting both PBRM1 and SMARC1, two other components of the SWI/SNF complex, was lethal, even though deleting either one was not. So these findings will show us together how this technology can map out dependencies that tumors have developed, which allows us to target them. So even though this field has all these technologies and studies that have been done, there are still some prominent gaps that have yet to be addressed. And one of them is being that most synthetic lethality screens will only measure viability, meaning that they're only measuring if a cell will live or die. But we need to understand how cells change in different ways through cellular differentiation, such as immune evasion, which will happen in a real body, in a real cell. So we need to study those apart from just a cell viability. And this is where something called single-cell RNA sequencing becomes useful, because it allows scientists to study how gene knockouts change gene expression in each individual cell. And a new model that has been developed, and similar models are also being developed, is the computational model DGI-B4SL, which is a predictive model. So it uses these neural networks and graphs to predict synthetic lethal gene pairs based on known gene interactions. And these models can help researchers and narrow down gene pairs that can be promising for experimental testing, because it's quite difficult to identify gene pairs that work together since there's thousands and millions of them. So this one, although it's still a predictive model, it can allow us to identify some. So for the conclusion, just to wrap up this paper, epigenetic synthetic lethality offers a new path for developing therapies that are both precise and specific to the tumor itself, rather than just harming healthy cells, which we want to avoid. And by understanding how these cancer cells become dependent on certain chromatin regulators through either gene loss or backup reliance, we can design treatments that selectively kill cancer cells. And technologies such as CRISPR and codependency screens allow us to systematically identify these synthetic lethal gene pairs. And the goal overall is to find these gene pairs, possibly test them in a clinical site, and develop therapies. So thank you. That was my presentation. I'm open for any questions.